So the last major anatomical structure we're going to discuss is the cerebellum. It's a very complex structure anatomically. It's got a thin outer layer of neurons that's vaguely similar to the main cortex, and also some inner nuclei, vaguely similar to the subcortical structures. And in addition, there's a couple of nuclei in the brainstem, the pontine nucleus and the inferior olive, that are closely interconnected with the cerebellum and can be regarded as part of the cerebellar system. So the cerebellum's located at the back of the brain. It's under the cortical hemispheres and hangs off the back of the brainstem. This picture shows an outside view of the brain, and if you strip away the cortical hemispheres and also the left hemisphere half of the cerebellum, you can see the folded outer layer of neurons surrounding an inner body of white axon matter. The folded outer layer is called the cerebellar cortex, and the folding is very convoluted, which gives a cross-section of the white matter the appearance of a heavily branched tree. The folded neuron layer in the upper brain is just called the cortex, and the folded layer in the cerebellum is always referred to as the cerebellar cortex. Sometimes, if it's particularly important to be completely clear about what we're talking about, we'll call the upper brain cortex the cerebral cortex. Very deep in the cerebellum, in the white matter, are some nuclei, the fastigial, the interpositors, and the dentate nuclei. If you cut a horizontal section of a human brain at the back, roughly where the arrow's pointing, it goes through the cerebellum. When the section's stained so that neuron somas appear blue, it looks like this. The cerebellar cortex has three lobes: the archicerebellum, the paleocerebellum, and the neocerebellum. And these three lobes are there in both hemispheres. In evolutionary terms, the archicerebellum is the oldest, and the neocerebellum is the newest, and the neocerebellum is much larger in human species than in any other. You can also see the cerebellar nuclei, the fastigial, the interpositors, and the dentate. And that's the pontine nuclei in front of the cerebellum in the brainstem. So that's what you can see in this kind of section, but if you make a sagittal section, which is a vertical section going from front to back and slightly offset from the center towards the left in this case, it looks like this, again with neuron somas stained blue. You can see the cerebellum, and as before, the pontine nuclei. You can see the inferior olive a little bit lower down. The white matter, circled in green on the left, is axons coming from the cortex to the pontine nuclei. This is called a cerebellar peduncle. The axons circled in green on the right are from the pontine nuclei into the cerebellum. It's worth noting that these axon bundles have around 20 million fibers. That's about 20 times as many as there are in, for example, the optic tract that carries information from the eyes to the brain. So clearly, a lot of information is going down from the cortex into the cerebellum. Now, as I just pointed out, there are three cerebellar cortex lobes, and there's also three major nuclei. And it's found that one lobe targets just one of the nuclei, and the different lobes and the corresponding nuclei have different types of roles. The oldest lobe in evolutionary terms plays a role in maintaining balance. The paleocerebellum plays a role in maintaining posture. What I mean by that is, if someone was to push your arm, the arm would automatically resist being moved. So there's muscle effort going into maintaining your body in the same position. And this muscle effort, called muscle tone, is directed by the cerebellum. Then the neocerebellum is involved in detailed control of voluntary movements, the ones our brain selects in response to our environment. But of course, in humans, some very tightly controlled mouth and throat muscle movements are involved in producing speech, and these movements are also directed by the cerebellum. And in addition, some higher cognitive processes that don't involve motor movements are also influenced by the cerebellum. But now let's have a closer look at the anatomical structure itself. If you take the region inside the rectangle and expand it, remember the neuron somas are stained in blue, so you can see the intricate folding of the layers. If you expand a small region of the layer even more, you can see there's a fairly broad band of neuron somas, following that green line. The neurons in this band are very small, so they've been labelled granule cells. And there's lots of them. In the human cerebellum, there's about 50 billion granule cells, compared with only 20 billion neurons in the whole cortex. And the total number of neurons in the brain as a whole is only about 85 billion. So almost 60% of the neurons in the brain are cerebellar granule cells, but they are very small. If you look even more closely, to the left of that broad band of granule cells, you can see another much thinner layer of much larger somas right next to the thick granule layer. And these somas belong to neurons called Purkinje cells. And you can call these three layers the inner layer granule layer, the middle Purkinje layer, and an outer layer called the molecular layer. The molecular layer contains mostly the axons of granule cells and the dendrites of Purkinje cells, although there are a few scattered neuron somas. If you pick a small region, like the one in the red rectangle, and enlarge that one, you can see the thick layer of granule cells on the right, the thin layer of much larger Purkinje cells in the middle, then the thicker molecular layer. I've circled one Purkinje cell in green. You can see its dendrite extending across the molecular layer. And these Purkinje dendrites have quite a striking shape. They're flat. In this picture, you can see it edge on. But if rotated, it would look like this. In other words, it's flat but extensive in 2 dimensions. The axons from granule cells go up through the Purkinje layer into the molecular layer, as I've conceptually illustrated in orange, and then they do a 90-degree turn and run perpendicular to the Purkinje dendrites. And each Purkinje gets 1 or 2 synapses from maybe 100,000 different granule cells. The somas of granule cells are very small, and their dendritic trees are also small and fairly simple. They have 3 or 4 short dendrites, and each dendrite gets lots of synapses from just one incoming axon. The multiple synapses give the end of the dendrite a vaguely claw-like appearance. And then there's some other types of neuron in the cerebellar cortex, but we don't need to discuss them at this point. In the cerebellar nuclei, there's 2 types of projection neurons. There's GABAergic inhibitory neurons that target the inferior olive, and glutamatergic excitatory neurons that target the thalamus and the spinal cord. These projection neurons fire tonically. In other words, they fire steadily in the absence of input, but the firing rate is modulated by their inputs. And there's also inhibitory interneurons with axons limited to within the cerebellar nuclei. An interesting point is that outputs from the cerebellum to other targets, other structures, only come from the cerebellar nuclei. The cerebellar cortex only targets the cerebellar nuclei. and I'll say more about this connectivity in the next section.